EpiBiome is a precision microbiome engineering company whose mission is to surveil microbial populations and to develop tools that help humanity combat antibiotic resistance. We have two main specialties. The first is bacterial profiling. So we can take a sample, it doesn't really matter where it comes from, and we can tell you accurately, usually to the species level, and semi-quantitatively what's in that sample. The second part of what we specialize in is specifically deleting single bacteria, problematic bacteria, leaving the rest of the community intact. Yeah, so FMT, or fecal microbiome transplant, is one approach whereby bacteria are introduced from a donor to a recipient to improve a condition like C. diff. Our company focuses on the use of bacterial viruses. So rather than bacteria or live biotherapeutics, we use viruses that are the natural enemies of the bad bacteria to specifically eliminate the pathogens. Yeah, there are lots of concerns with using phages. There's no precedent for an FDA-approved phage therapy in humans or in animals. That doesn't mean that it's not possible. It just means that it hasn't been done yet. Phages have been around for the last 100 years. Some of the challenges that we face are things like manufacturing. You know, this isn't a small molecule, so a lot of the methods of GMP manufacturing of a small molecule have to be changed a little bit for phages. All of this can be done, but again, it introduces more cost and some uncertainty, and sometimes just paving the way to do something new takes a little bit more time and money. So that's one aspect. Now remember, antibodies as a therapy would have been preposterous if you had asked people that 20 years ago. But today, they're quite mainstream. So, I think anytime you have a new paradigm, it just takes time and money. Other challenges with phages are host range. So these are bacterial viruses that have a spectrum. Some of them are very specific for particular bacteria, and some of them are actually quite promiscuous. So they will infect all of the strains in a particular species, sometimes even crossing the species level to hit multiple species within a genus. So a lot of developing a cocktail that works well for addressing the pathogen, many of these considerations have to be taken into account. Yeah, so as I mentioned, these bacterial viruses have a spectrum of host range. What we can do is we can tailor the phages that we use in the cocktail and choose phages that tend to be quite specific for the pathogen. So one strategy that we really like is taking a pathogen with a receptor on the surface, and that receptor is somehow important in the pathogen's ability to cause disease. And then we can target the phage to that receptor. And the idea is that if the bacteria gains resistance by loss of the receptor, now the bacteria can't be killed by the phage, but it is also no longer capable of causing disease. And that's a really profound concept. It's not something that you can do with small molecule antibiotics today. You're basically forcing bacteria to choose between survival or the ability to cause disease. That's a really neat concept. Yeah, so our two programs today, we have one in human health. We're tackling environmental enteropathy. It's caused by chronic infection of the gut. It disproportionately affects infants in developing countries. These infants, through their food supply, are getting contaminated with fecal material. Some of these bacteria latch onto the gut lining of the infant, secrete an enterotoxin, which causes massive inflammation. The infants stop absorbing nutrients. They show stunted growth, and they stop responding to oral vaccines. Now, as you can imagine, the Gates Foundation cares a great deal about this lack of response to oral vaccines. So this is one exciting project that we are partially funded by the Gates Foundation to work on. The second project is in animal health. So we're targeting a disease called bovine mastitis. It's an inflammation of the udder tissue in dairy cows, usually caused by infection. And it's the #1 reason to give a cow antibiotics today. So we are actually replacing those existing antibiotics with our phage therapy. And we have some great data in a 20-cow study. Yeah, so when we think about a single approach, I don't really believe that any single approach is going to be the solution to all of humanity's problems, bacterial problems. I think it's really going to be a combined approach. I think small molecule antibiotics will still play an important role. I think you'll have an important role from lysins, from bacteriocins, natural antibiotics that bacteria secrete, from bacterial viruses, phages, and even live biotherapeutics. So it's really going to be this combination approach. Some of these tools will work better for certain applications. Some of them will work really well together. And this may require developing new regulatory pathways. Today, these sorts of combination therapies are practically impossible to get approved just by virtue of what you have to show to FDA in terms of synergy. So I'm excited about the future. I think that there are applications well outside of human and animal health. I envision applications in oral care, skin care, aquaculture. I think you've got pests even. You can target their microbiome and kill pests, like termites, for example. All sorts of neat opportunities, and we're really just on the cusp of learning how to deploy these tools to tackle some of the problems that we've previously been tackling with antibiotics or chemicals.